Hi everyone. Have you ever wondered how the butterfly came to be? Or how its amazing life cycle came into existence? If so, you are in the right place. Because in this presentation, I'm going to talk about metamorphosis and the origins of how a caterpillar turns into a butterfly. We are going to dive deep into the biology, the evolutionary story, its difficulties, and what that implies for the origins of complete metamorphosis. By the way, this presentation is based on Chapter 3 in my book, Amazing Insects: Evidence of Design, chapter titled The Astonishing Transformation: Complete Metamorphosis. This book is available online for just $10 via the QR code shown on this screen. Thank you in advance for your support. There are three main types of insect life cycles. First is simple growth, where the egg hatches into a miniature version of the adult. This is the case, for example, with silverfish, jumping bristletails, and firebrats. Insects have hard skin, called an exoskeleton, which restricts how much they can grow. To get bigger, an insect must shed its hard exoskeleton and allow the new, larger one that developed underneath to harden. This process is called molting. The stage between molts is called an instar. The second type of insect life cycle is incomplete metamorphosis. In the usual case, an egg hatches into a nymph, which looks like the adult but has immature adult features that need more development. In some cases, the nymphs have a different lifestyle in terms of habitat than the adult. The young spend their lives underwater before undergoing the final molt to become a winged adult. While this is still a remarkable transformation, it pales in comparison to the third type of insect life cycle. The third type is called complete metamorphosis, which is a total architectural remodeling, not just simple biological growth. Let's start with the egg. This is a self-contained unit engineered with 100% water-resistant, non-toxic, heat-resistant adhesive glue. The larva, in butterflies known as a caterpillar, is an eating machine. It increases its weight by up to 3,000 times, which is equivalent to a 6-pound human baby growing to an 18,000-pound elephant in a few weeks. A caterpillar molts several times, allowing it to get bigger. In the monarch, there are 5 larval molts and instars. The pupa, in butterflies known as a chrysalis, is the transformation chamber where the caterpillar turns into the adult butterfly, which is specialized for flying and reproduction, not growth. Now, let's dive deeper into how a caterpillar transforms into a butterfly. We first need to understand that there are 3 chemical architects or hormones that dictate the timing and nature of every phase in the butterfly's life cycle. First is PTTH, the initiator. It's released by specialized brain cells that monitor growth. It acts as the chemical messenger traveling through the circulatory system to trigger the molting sequence. Second is ecdysone, the builder. It's released by the prothoracic glands. It is the primary molting hormone that triggers the complex sequence of shedding the old exoskeleton and forming a new one. The third is juvenile hormone, JH. This is present during the larval instars, and it dictates the outcome of the molt. High levels ensure the skin cells produce just another larval exoskeleton, locking the insect in the growth phase. To start illustrating this, look at the top right, where the brain releases PTTH. This stimulates the prothoracic glands to release ecdysone when it is time to molt. When ecdysone is released, one of 2 things can happen depending on whether juvenile hormone is high or not present. If juvenile hormone is high, the caterpillar molts to just a larger caterpillar. But if juvenile hormone is basically 0, this triggers the caterpillar to start getting ready to pupate. The chart on the left is not completely accurate, so let me show you a better one that describes what really goes on. This is really a more accurate chart of how juvenile hormone and ecdysone interact from hatching to adult emergence. The horizontal x-axis represents time, which is not the scale, as the monarch butterfly develops. The vertical y-axis represents the amount or concentration of hormone. The hormonal control of monarch development is a 3-act drama. In Act 1, we have high juvenile hormone plus periodic ecdysone peaks. Every blue ecdysone spike triggers a molt. Because juvenile hormone stays high, the insect just grows into a larger caterpillar. So from instars 1 to 4, juvenile hormone acts as a larval shield, preventing metamorphosis. The second act kicks off with instar 5, or the larva 5, L5. Here, the larva hits a critical weight and juvenile hormone plummets. This removes the shield and signals the start of the next phase of metamorphosis. There is a small blue ecdysone pulse, which triggers the commitment to metamorphosis gene program, followed by a brief prepupal juvenile hormone surge, which acts as a safety mechanism, ensuring the insect forms a pupa rather than skipping straight to an adult, which would end up malformed. Then there is a final sharp blue pulse of ecdysone that triggers the actual transition into the chrysalis. Once inside the chrysalis, Act 3 begins. This is the adult differentiation stage. Here, a massive sustained ecdysone mountain, essentially, drives the complete cellular reconstruction of the adult body. Once the adult is formed, hormones must drop to 0, and this quiet period allows the exoskeleton to harden, tan, and cure properly, preparing the butterfly for eclosion or emergence. Now we've seen how juvenile hormone levels change over the life cycle and established that high juvenile hormone levels act as a perpetual brake on adult development. But what does juvenile hormone actually do? How does it work? Well, high levels of juvenile hormone maintain the expression of a critical transcription factor called cruppal homolog 1, KRH1 for short. This is like a lock on the DNA, preventing the expression of a gene called E93. The E93 gene is necessary for adult development. As long as KRH1 prevents the expression of E93, the genetic blueprint for making wings, compound eyes, and other adult structures remains entirely suppressed, allowing the larva to not waste resources or energy on things needed later on, but allows the caterpillar to focus purely on rapid feeding. Now, to further complicate things, there is another factor that we need to consider, and that is the broad complex, or BRC, protein. When ecdysone surges a small amount, the low blue bump, it signals the caterpillar to get ready to pupate. Juvenile hormone is basically at 0. So when this happens, ecdysone activates BRC instead of KRH1. During the late larval and prepupal stages, one of BRC's jobs is to repress the adult genetic program led by the E93 gene, which should only be activated once metamorphosis is underway inside the chrysalis. KRH1 formerly repressed E93, but BRC serves as a substitute for KRH1 in blocking the expression of the E93 gene in the absence of juvenile hormone. So, high levels of BRC keep E93 turned off. This keeps the insect in its adolescent or pupal commitment phase, ensuring tissues develop properly before transitioning to adult differentiation. Once the insect transitions fully into metamorphosis in the chrysalis, the regulatory dynamic flips dramatically. E93 represses BRC. As the large ecdysone pulse triggered the next phase, E93 expression surges. E93 directly represses the expression of BRC. But BRC doesn't just repress E93. BRC acts as an incredibly versatile multi-tool, coordinating 3 other essential pupil developmental subroutines. The second function is imaginal disc development. BRC unlocks and coordinates the rapid proliferation and elongation of internal wing, leg, and antenna packets through the imaginal disc. Third function is controlled demolition or histolysis. BRC triggers programmed cell death, apoptosis, and autophagy, systematically dismantling larval salivary glands and crawling muscles. The fourth function involves chrysalis formation. BRC-specific protein isoforms Z1 and Z4 downregulate soft larval cuticle genes while upregulating the specialized genes required to build the hard chrysalis shell. Now, when you consider the regulatory circuits for metamorphosis involving juvenile hormone, ecdysone, KRH1, BRC, and E93, we can identify 3 major stages from larva to adult. In stage 1, larval growth, high JH and KRH1 repress BRC and E93. But what if JH and KRH1 were absent? Nothing would stop the activation of the adult genetic program early on, and you would get a tiny little larva trying to turn into an adult before it was ready, a lethal proposition. In stage 2, pupil commitment, juvenile hormone drops off, and the ecdysone pulse without KRH1 activates BRC, which represses E93. But if BRC did not exist, you would get premature E93 or adult genetic program activation, completely skipping the pupa. The caterpillar would try to turn directly into a butterfly without forming a chrysalis or going through the necessary pupal steps. This results in catastrophic developmental chaos, leaving the insect trapped in a severely deformed, half-larval, half-adult state, called a teratological monster, which cannot survive the final molt, if the caterpillars survive long enough to permit even that. In stage 3, for the adult transition, ecdysone surges activating E93, which represses BRC, leading to the formation of adult tissues and organs in the chrysalis. But what if there was no E93 gene? Well, there would be no adult program activated, and the pupa would remain trapped in a goo-like state in the chrysalis, which is obviously ultimately fatal. Now, let's dive deeper into the evolutionary story. Let's start at the beginning with the egg. There is an embryo inside the egg, which at first is just a bunch of cells. At first, there is no juvenile hormone, and as the cells multiply, there is a patterning process that takes place, mapping out where different body parts are going to develop. At a certain point, there is a surge of juvenile hormone which turns off the patterning and triggers differentiation and maturation of tissues. This marks the beginning of the pronymph, what the embryo turns into inside the egg before hatching. Now, evolutionists believe that holometabolous insects like butterflies evolved from hemimetabolous insects that grow and mature through multiple nymphal instars that with each molt get closer and closer to the adult phenotype. Examples of hemimetabolous insects include cockroaches and crickets. These types of insects lack a pupal stage. So normally, after patterning is complete, in the middle to late embryo stage, the glands that make juvenile hormone wake up and flood the embryo with juvenile hormone. But evolutionists propose that something unusual happened once upon a time. The pro-nymph hypothesis suggests that a mutation made juvenile hormone to start being produced early, in the middle of the normal patterning process, prematurely forcing the embryo to rapidly differentiate and mature whatever structures were mapped out at that time. The hypothesis proposes that such structures like advanced compound eyes, long legs, and mature wing pads were not mapped out yet. Ultimately, this is all thought to have caused the pronymph to hatch early, creating the first free-living, soft-bodied, simplified larva, which evolved and adapted to become like a caterpillar. Now, if you give a construction crew building a house a blueprint, then take away the blueprint before they have finished mapping out on the ground where things are going, and tell them to just build a house with what is mapped out already, you're going to have a partially constructed house, perhaps missing certain walls, or kitchen countertops, or toilets, etc. You see, you can't just get a mutation affecting hatch timing and get a free-living larva. Such a mutation could only cause an incomplete hemimetabolous insect to emerge from the egg, if even that, but it would probably prove fatal because, along with the change in juvenile hormone timing, there would have to be a simultaneous rewiring of the downstream gene regulatory networks, or GRNs, controlling how they respond to the shift in juvenile hormone so that larval features are activated appropriately. If not, chaos would ensue. So, from an intelligent design perspective, we see a need for completely new yet coordinated blueprints outlining the specialized larval or caterpillar, pupa, and adult stages. A caterpillar is significantly different from an unfinished nymph that hatched prematurely. Evolutionarily, if the supposed early hatching pronymph managed to successfully hatch out of the egg, it would need certain larval features to survive. At minimum, the necessary adaptations needed would include, first, hard external mouthparts ready to chew. Second, digestive tract ready to process food from the mouth, not the yolk sac. Third, crawling prolegs under the rear body to prevent major damage from dragging a soft body along, which would also require their associated musculature and nerve connections. Fourth, defense mechanisms to protect against predation would need to be present, whether that is camouflage, deterring colors, toxins, whatever, along with defensive behaviors. But that is not all. Actually, the evolutionary story requires at least 6 massive developmental shifts to occur. We just talked about the first one, the hormonal shift. The first 3 shifts constitute what I call the triple genetic leap, which had to have been made by the first early hatched larva to survive on day 1. Building on the premature hormonal shift of leap 1, leap 2 addresses the radical structural transformation required to turn an unhatched embryo into a functional mobile larva. Normal nymphs rely on an enzyme called lactase 2 to chemically harden their outer skin into a protective armor plating, using localized muscle bundles attached to internal skeletal pegs, like a system of mechanical levers. A larva, however, requires a completely inverted blueprint. It must suppress lactase 2 and upregulate RR1 cuticular proteins to create a soft, highly elastic skin. However, in order to move a soft body, the insect needs a different muscular framework, specifically continuous, intersecting sheets of longitudinal, oblique, and circular muscles. The ultimate result is a pressurized hydrostatic skeleton where muscle layers contract uniformly against internal fluid, called hemolymph, using the stretchy RR1 cuticle as a structural container. Leap 3 is sensory behavioral gene regulatory network activation. You see, even if an early hatched insect miraculously acquires a perfectly pressurized hydrostatic body, it remains behaviorally dormant and blind, trapped in a deep embryonic sleep. Leap 3 requires an immediate, simultaneous rewrite of the insect's neural gene regulatory networks, or GRNs, to instantly adapt its mind to the outside world. Without this sudden neurological awakening, the premature hatchling would simply lie motionless and starve to death. So to become a viable, self-sustaining eating machine, the insect's brain must instantly activate functional olfactory, or smelling, and gustatory, or tasting, receptors to recognize and locate host plants to eat. At the exact same time, it must switch on positive phototaxis, that is, the instinctual drive to move toward light, which is where leaves are found, and activate the central brain circuits required to coordinate hydrostatic crawling and mandibular chewing. To try to help you understand the implications of the triple genetic leap better, let me try to restate each of the leaps for you, focusing on the predicted outcome if the shift failed to occur, as well as what genetic information is necessary. For leap 1, forcing a premature hatch by disrupting the embryonic clock triggers lethal developmental chaos, leaving an unfinished embryo to run its default nymph program. Much like snatching a blueprint away halfway through framing a house, this developmental disruption results in a structurally compromised, non-functional dead end. To survive outside the egg, the insect completely lacks the necessary simultaneous gene regulatory network rewrite for larval features to replace the interrupted nymphal blueprint. Without the immediate coordinated appearance of hard mandibles for chewing solid food, a modified digestive tract to process it, and vital defense mechanisms such as camouflage, warning coloration, or toxic chemical defenses, the pronymph remains an unfinished nymph doomed to starvation or predation. For leap 2, transitioning to a soft body without a total muscular overhaul results in a lethal mechanical catastrophe. Traditional localized muscle bundles, programmed by the gene MEF2 to anchor to a rigid lactase-2 hardened exoskeleton, would have no mechanical leverage. Instead, they would pull against a squishy body wall, causing the insect to buckle, crumple into a ball, and suffocate. Resolving this biophysical deadlock requires an upside-down rewiring of nymphal tissue systems, the simultaneous suppression of lactase-2, and upregulation of elastic RR1 cuticular proteins to create a flexible container, combined with a re-engineering of MEF2's downstream genetic targets to construct continuous intersecting muscle sheets. Without both components forming a functional hydrostatic engine on day 1, any gradual intermediate, such as a soft cuticle without hydrostatic muscle sheets, results in a total structural failure. It all has to be in place on day 1. Evolutionary theory attempts to evade this with an armored, running Campodeiform ancestor, but this merely kicks the can down the road, delaying the exact same mechanical trap down the timeline. For leap 3, even if a functional larval body could miraculously form, hatching early without a cognitive awakening results in lethal behavioral dormancy. Inside the egg, the pronymph is behaviorally blind and programmed for embryonic sleep. Without a sudden neurological awakening, the premature hatchling would simply lie motionless and starve to death. Materialistic frameworks cannot explain the highly specific neural genetic regulatory network modification required to activate a complete behavioral suite at the exact moment of hatching. To forage successfully, the insect must instantly express functional olfactory and gustatory receptors to identify host plants, as well as switch on positive phototaxis to navigate towards light, and fire central pattern generators in the brain to coordinate hydrostatic crawling and chewing, which is an all-or-nothing neurological package that cannot be built step by step. When you put the implications of the triple genetic leap together, we find that a failure in any single part of this endocrine, structural, or behavioral triad would result in death for the pronymph. Therefore, the entire multi-system genetic rewrite must occur and function perfectly on day 1. This is simply biologically impossible to achieve via a gradual step-by-step pathway. Now, I mentioned there were 6 difficulties that evolution faced. The fourth is somatic suppression and polyploidy. Under the neo-Darwinian framework, for a primitive insect to simultaneously halt adult development while driving massive larval growth, random mutations would have had to force the KRH1 master control factor into a flawlessly synchronized dual role. First, it would have had to achieve a developmental freeze by physically locking down and silencing adult-specifying genes like E93. But second, it would have had to simultaneously execute a radical cellular rewrite, forcing certain larval tissues to grow not through cellular mitosis, but through something called polyploidy, where one cell repeatedly copies its DNA without splitting into 2 cells. Now, the purpose of this is to allow for dramatic growth without getting bogged down metabolically by having to create all the components required for new cells and go through the complex process of mitosis over and over again. Polyploidy allows the larval body, especially the skin cells for example, to become a rapidly expandable soft cylinder designed for rapid feeding and growth. For this massive cell expansion to be viable, unguided evolution would have had to construct a highly precise, self-resetting molecular clock to safely drive this DNA copying without permanently freezing the cell mid-cycle. Furthermore, the organism would have required an immediate genetic override of ultrasensitive cellular alarms that normally command cells to self-destruct when they balloon with extra copies of DNA. Because any unguided step-by-step intermediate would have suffered either fatal cellular freezing, uncontrolled tumor-like tissue overgrowths, or catastrophic cell death, this independent dual control system would have had to emerge perfectly intact on day 1, an all-or-nothing masterstroke that completely defies gradualism. Difficulty 5 involves imaginal disc. To safeguard the delicate blueprints for future adult structures, like the wings, adult legs, and antennae, from the constant physical hazards of burrowing through abrasive soil or navigating dense, rigid vegetation, the evolutionary narrative requires that insulated internal sanctuaries, known as imaginal disc, would have had to materialize suddenly without any evolutionary precursors. While the surrounding larval tissues were held in a developmental freeze, highly localized clusters of embryonic cells would have had to achieve a precise escape by selectively turning off the KRH1 lockdown so they could multiply rapidly. Rather than a passive accident caused by crowded cells, gradual evolution would have had to seamlessly integrate 3 sophisticated genetic subroutines from day 1. Mapping out exact spatial grid lines across the tissue using chemical gradients like wingless and DPP, activating internal molecular motors to narrow the tops of specific cells like a drawstring purse to mechanically force the skin layer to buckle and pocket inward, and transforming this pocket into an insulated protective membrane sac. Without all of those 3 subroutines operating flawlessly from the very beginning, any hypothetical intermediate would have resulted in uncontrolled tumor-like cell overgrowths or fatal ruptures in the larval skin, demonstrating a structural reengineering that step-by-step pathways are completely unequipped to account for. Difficulty number 6 concerns the pupa and systemic dissolution and reconstruction. For a lineage of insects to successfully cross the developmental chasm of the pupa, gradual evolution would have had to perfectly coordinate an abrupt drop in juvenile hormone levels to remove the KRH1 developmental lock without triggering immediate biological chaos. The evolutionary mechanism would have had to instantly reassign master control to the broad complex protein to interact with E93 and flip the pupate switch. This switch would have had to concurrently orchestrate systemic dissolution, histolysis, and reconstruction, histogenesis. Now, if that was confusing to you, I'll try to explain in more depth in just a bit. In the supposedly ancestral hemimetabolous insects, BRC has a completely different regulatory function, that of controlling cuticle growth and wing pad development in nymphs. But the evolutionary story posits that over a long time, the BRC gene just so happened to become the master pupil regulator in insects that evolved to mature through complete metamorphosis, holometabolous insects. The story is one of co-option. Over time, the BRC gene sequence didn't change, but the downstream genes, cis-regulatory elements, that it targets for regulation gradually changed, favoring those that first help the insect to rest during molting, then minor remodeling genes changed to be regulated by BRC, until finally genes controlling histolysis, histogenesis, and pupation were fully captured, enabling complete metamorphosis. Now, we are talking about thousands of genes that before were not regulated by BRC, all acquiring highly specific coordinated binding sites for BRC. That is a lot of genes. Now, the critical flaw with the evolutionary narrative here is that if you don't have the necessary genetic information to complete the pupation and form a functioning, reproducing adult, it is a dead end evolutionarily. While you can gradually change how a nymph molts into the next instar, you cannot gradually change how a caterpillar turns into a butterfly. That's a whole different program. If you change a master factory command, step 1, to make the assembly line stop and go quiescent or rest, but you haven't yet written the software for the automated machine to do anything new, well, you've just broken your production line for no reason. On the flip side, if you skip ahead and add powerful new demolition or rebuilding tools to the factory floor, steps 2 and 3, without an incredibly sophisticated, perfectly timed master computer program already in place to control them, those tools will fire off, maybe at the wrong time, and destroy the entire factory during a routine maintenance cycle. Now, evolutionists try to get around this by pointing out that an insect's DNA is organized into a hierarchy, like a CEO giving orders to a manager, who then commands hundreds of factory workers. They argue that evolution only had to change the master switch at the very top, the BRC gene, to take control of everything below it. But this actually compounds the problem. Altering a top-level master switch forces an entire pre-existing genetic network to fire prematurely, causing lethal developmental chaos rather than gradual progress. You see, a master switch, like BRC, cannot become the master pupil regulator one gene or even one group of genes at a time for the same reason that a half-written computer program doesn't run halfway. It simply crashes the system. This creates an impossible mathematical bottleneck known as the waiting time problem. For a completely novel metamorphic stage to successfully emerge, the master commands and the downstream execution software must be reprogrammed concurrently. Waiting for unguided mutations to accidentally write multiple perfectly coordinated pieces of complex software at the exact same time would probably take billions of years, resulting in a lethal dead end for the insect even over deep time. Now, let's discuss the necessary novel hardware for pupation. On the left, the hemimetaboly system, represented by the cricket, possesses a core toolkit of conserved insect genes responsible for basic cellular growth, signaling, and localized tissue remodeling, as seen by the Wnt, hedgehog, caspases, and ATG genes. However, its developmental life cycle completely lacks a pupal stage. Its genome lacks the coding material entirely for specialized pupal structures, meaning that a simple shift in developmental timing cannot explain the origins of the complex metamorphic life cycle. There had to have been a massive influx of entirely new gene families that do not exist in incomplete metamorphosing ancestors. To achieve a viable complex pupal stage, the genome must possess silk production genes for cocoon construction, specialized pupal cuticle proteins, and cross-linking enzymes to build a rigid protective chrysalis, and number 3, a highly synchronized prophylactic gut immunity subroutine to sanitize the digestive tract right before it breaks down in histolysis, preventing any dangerous gut microbes from taking over. That in itself is amazing and shows foresight. How would an evolving caterpillar know it needed to sanitize its gut before dissolving it? From an intelligent design perspective, this blueprint demonstrates that complete metamorphosis relies on the simultaneous presence of novel structural building blocks and systemic subroutines, a feat of genetic engineering that cannot be achieved through gradual shifts in juvenile hormone timing alone. All this brings up an important point about evolution. The evolutionary engine requires uninterrupted reproductive viability. Neo-Darwinian evolution rests on a three-legged stool. If one leg fails, the entire process collapses. The heredity bottleneck. Natural selection cannot act on non-viable or non-reproductive intermediates. If a genetic mutation creates a beneficial larval trait, but that specific organism cannot successfully reach adulthood and mate, the mutation dies with it. There is no biological foresight to save a half-evolved mechanism for later generations. We can illustrate this through the vanishing bridge of heredity. The way complete metamorphosis works, the insect enters a transiently non-viable stage called pupation. It cannot reproduce while in the chrysalis. In order for the caterpillar to reproduce and thus pass on any potentially beneficial mutations, it has to cross the bridge to an adult butterfly with sexual organs that can mate with an adult of the opposite sex. If pupation fails, no adult, no offspring, no bridge of heredity. You cannot cross the bridge of heredity by slowly constructing the pupal phase over many generations. This is impossible. Natural selection cannot preserve a partially evolved pupa. The bridge vanishes unless the entire transformation program is present and perfectly synchronized from the start. But an evolutionist might point out that this is a straw man. For complete metamorphosis developed from ancestral insects that had quiescent pupa stages, like thrips or whiteflies. Well, these insects do have a bit of an anomalous life cycle, in that after the second larval instar, the third larval instar is a propupa, and after the fourth molt is a pupa. The propupa and pupa undergo a semi-quiescent resting phase and are non-feeding. If poked, they can awkwardly crawl away, but they do not actively move or seek out food. The main difference, though, is mechanistic. In a thrip, the propupa and pupa stages are merely a behavioral and hormonal pause. Internally, their anatomy remains continuous with gradual tissue remodeling of the muscles, gut, and nervous system, etc., to fit the adult form. They do not undergo complete system-wide histolysis, histogenesis, or have imaginal disc. Thus, although thrips do represent a form of crossover between hemimetabolous and holometabolous insects, they are still firmly on the side of hemimetabolous. The insect's anatomy, especially internally, remains largely continuous between the larval and adult stages. There is a profound chasm in terms of tissue reorganization between thrip-like insects with semi-quiescent pupa and true holometabolous insects, where the guts, muscles, and nervous system are dissolved into a metabolic soup. A death sentence unless the program for controlled dissolution and reconstruction from imaginal disc is fully complete and operational. So when it comes down to it, there are two explanatory frameworks to interpret the data we are seeing. Either complete metamorphosis evolved via a blind process, neo-Darwinism, or it is the product of intelligent design. On the one hand, we have undirected, random genetic mutations seeking immediate survival advantages. On the other hand, we have pre-programmed, interconnected developmental pathways designed for a final goal. How do these two perspectives handle the pupal chasm? Well, neo-Darwinism requires a biologically impossible scenario where natural selection builds a non-reproductive, non-viable transient stage step by step. Intelligent design accounts for the chasm by preloading the exact instructions into the DNA from the beginning. Where does the information come from? Neo-Darwinism postulates that the innate knowledge, like when to hatch, when to molt, and how to knit a proboscis, all arose by chance, without empirical confirmation. But no scientific study can prove this. Intelligent design posits that a mind capable of purposeful creation wrote the complex blueprint into the DNA before the first life cycle ever began. I believe that the sheer wonder and intricate solutions of insect metamorphosis point in the direction to the ingenuity of deliberate design. A system demanding complete integration from the outset points to deliberate design. The problem with blindness. Evolution lacks inherent direction. It does not know that a non-viable, immobile pupal stage must eventually produce a reproductive adult. The implicit requirement. An organism essentially enters a transiently fatal developmental stage with the implicit requirement of emerging successfully. The necessity of a plan. The ability to foresee the entire life cycle, integrating the precise hormonal timers and the triple genetic leap to cross the vanishing bridge, strongly suggests a mind capable of purposeful creation. The emergence of the flying, reproductively capable adult is the culmination of flawlessly executed innate capabilities, present from the moment the egg was laid. This is beyond random chance. The astonishing biological programming required to orchestrate complete metamorphosis reveals an inherent resilience, a profound ingenuity, and undeniable purpose. So, in conclusion, it is a process that speaks not of blind accidents, but of the magnificent forethought of an intelligent designer. With that, I'd like to thank you for your attention during this presentation. I hope I have given you much to ponder and wonder at. If you would like, feel free to drop a comment below, or head over to my website linked in the description, lessonsfromnature.blog. Thank you for watching.